case of nucleic acids, right, what are the things we must know? So the most important component of nucleic acid is nitrogenous base. In fact, when it comes to nucleic acid, right, it would have a sugar which would be a pentose sugar, especially ribose, correct? And the ribose can be deoxified and we can get deoxyribose also. So when it comes to nucleic acids, one we will have a sugar which would be either ribose or deoxyribose. Next we will have a phosphoric acid, right? So that phosphoric acid derivative would appear like a phosphate ester. The third we will have is a nitrogenous base, correct? And this nitrogenous base would usually be heterocyclic in nature, correct? So first we will focus on pentose sugar, correct, which is either ribose or deoxyribose, and then we will discuss about nitrogenous bases and phosphates. Okay, let's see this. Ribose by structure, we all know that this is the Fischer projection of ribose, all OH on one side. Okay. Now, if at all I take the cyclic structure of ribose, correct, then this is how it appears. You can see this is the beta ribofuranose, got it. Now, check it carefully. This OH on the first carbon is up, so which suggests it is a beta ribofuranose. If OH is down here, it is called as alpha D ribofuranose, correct. And generally in nucleic acids we will see beta is most found as compared to that of alpha. Okay. Now coming to deoxyribose. Now observe this structure, cyclic structure of ribose. In this cyclic structure of ribose, suppose if we take this beta D ribose, got it, same beta D ribose except on the second carbon. Check out carefully. This on the second carbon, if at all I replace that with hydrogen, that means it got deoxified, so we call it as beta D deoxyribose. Okay, so that is about ribose and deoxyribose. Now let us focus on the nitrogenous bases, especially the heterocyclic part of it. When it comes to nitrogenous bases, it would have two parts. One is a purine derivative, the other comes from pyrimidine derivatives. Got it? Purine would have two rings here. One is a this is a pyrimidine ring and this is a pyrrolidine ring. So both combined together will give us purine ring. When it comes to pyrimidine, pyrimidine is this regular one which is, you know, if you have two nitrogens, we will call it as pyrimidine. If you have only one nitrogen, we call it as pyridine, right? Now, if you observe these two, let us just focus on purine once and see what would happen here. So, in case of purine, we can get two bases, two nitrogenous bases with the same structure. So, this is a basic purine structure. So, on this purine structure, if you have NH2 on this sixth carbon and here NH2 on the second carbon and OH on the sixth carbon, we will get adenine and guanine. I guess you understood that. We will get this as adenine and guanine. So these are the two nitrogenous bases which we will be found in nucleic acids. Now if you observe this carefully, on sixth carbon, this is actually an enol form which exists in a keto form like this, which is amide form, correct? So that would be amide and enol kind of a form. You can see that. So guanine will have this enol kind of a form, right? So this is with respect to purine. Now let us see the derivative of pyrimidine. So from this pyrimidine structure, how many derivatives we can get? So if you take this pyrimidine, we get three derivatives of this pyrimidine. So if you see the first one, the first derivative will have NH2 on the sixth carbon, OH on the second carbon, okay, and this is called cysteine. And observe carefully on the second carbon, the double bond which is an enol form, now it has converted into double bond O. Okay. So, which is the cytosine formation, not cysteine, sorry, cytosine formation. Now, here if you see the second base, right, on 6th carbon as well as on the second carbon, both will be enol forms, and those enol you can guess they will convert into, you know, amide form now. So, you can see double bond O is formed here and double bond O is formed. This is called uracil. Imagine the same uracil here also. Everyone check out if there is a uracil formation here. Got it? Then also I will have the same problem. You will get the same uracil with these amide forms, but except on the 5th carbon, if you have a methyl, you call it as thymine. So, these are the 3 bases you will get there, which is cytosine, uracil and thymine. So, with purine we got 2 bases, with pyrimidine we got 3 bases. Here you have CUT, cut, okay. So, cut bases come from pyrimidine and AG, adenine and guanine comes from purine. I hope you can remember it that way, okay. Next, third one is a phospho ester which is present in the nucleic acids. If you observe, this is phosphoric acid. If the phosphoric acid is being made an ester form, suppose if you check out, one H has been replaced with R, then this would become phosphomonoester. If you remove 2 of them, it will become diester. If you remove 3 of them, it will become triester. Okay. Now, in this, if you observe, nucleic acid contains only diester. Most of the time, all the nucleic acids will contain phosphodiesters. Now, let us see what is nucleoside in this. See, the combination of this nitrogenous base and a sugar would make us a nucleoside, which means whatever the base we have just discussed, that plus sugar, if you combine both of them, we will get this as nucleoside. Okay. And the sugar may be either a ribose form or in the deoxyribose form. Okay. Now, important point is the combination of this nitrogenous base plus sugar, which we just called it as nucleosides. If at all we also connect that with phosphodiester, that would become nucleotide. Okay, the side will become tide. Please remember that the side will become tide. So, all the nucleic acid will have a combination of nitrogenous base, a sugar and a phosphodiester, and that is nucleotide. Okay. So, you can see that here base plus sugar, which is nucleoside, plus phosphate will become nucleotide. Okay. Now, nucleoside, which suppose if you check out the constituent of adenosine, adenosine is a nucleoside. And how it is formed, let me show you. Suppose if I take beta D ribose, you can see the structure, this is beta D ribose, that if you connect with adenine, see how I am connecting here, on this nitrogen H and OH from this first carbon, if you remove the water, there is a connection that is established, and that connection is a nucleosidic connection, and that combination is called as adenosine. Now, it is a combination of adenine and ribose, so that's why it is called adenosine. Okay. Similarly, if you check out deoxyadenosine, deoxyadenosine again will take the 2-deoxyribose. You can see on second carbon, if you have H, we call it as deoxyribose. That if you connected with adenine, same on the 9th carbon, you can check out 9th nitrogen, you can check out if the connection is established this way. It is a combination of adenine and ribose, so which will become deoxyadenosine. Okay. So, this is a important question that has been asked in JEE or in fact other competitive exams. What is an adenosine? What is a deoxyadenosine? Okay. Next, if you check out nucleoside, now this is a nucleoside adenosine. How does it get connected to form, you know, nucleotide? So, now we can see if you check out the structure here, right, on this carbon. The fifth carbon, this CH2OH, I should be able to represent this way, okay, and on the third carbon OH is anyway down. So observe that carefully, this becomes very useful for us, okay, considering this 2-deoxyadenosine that can also be represented this way. Now without H, I don't have to show again H because these two hydrogens would be present here anyway, okay. Now these are the representations. Let me repeat once again. So this is a representation of adenosine and this is the representation of 2-deoxyadenosine. Taking these two, now we can make nucleotide, okay, C, which is a combination of base, sugar and phosphate, correct? Suppose if you take adenosine, 5 what do you say uh, 5 bar monophosphate, which is a ribonucleotide, got it? How does it look like? See that, okay. So here we have, this is a see OH is present here, so this is actually a ribonucleotide, not deoxyribo. So this is a nucleoside and this is the phosphate. Now we are trying to combine these two to get a nucleotide linkage. Check out carefully now. So this OH and OH has lost, so phosphate got connected here. So what has just formed is the nucleotide. So I hope all of you can make out what is nucleotide, right? So the formation of 2-deoxyribose, right? If you check it out, in the second carbon we have both hydrogens, which means it is deoxyribose, and with connection with this we will get an connection here on third, and first we will get an linkage which is nucleoside. Now it has formed nucleoside. Everybody got this? This nucleoside, if at all I have to make it as nucleotide, I will take these two and connect with phosphate. Got it? So where do we connect now? On the third carbon here, if I can connect with the phosphate, now this also has become a nucleotide form. Got it? So what is the name for this? 2-deoxycytidine, right? 3-monophosphate. So that would be the nucleotide which has formed. And this in fact is a deoxyribonucleotide. Okay, so this is a deoxyribonucleotide. Okay, I guess that is clear. Now if you check out DNA and RNA, in DNA what you would see, nucleotides are joined either through 3 bar or 5 bar or 3 dash or 5 dash phosphate linkages to give polynucleotides, which further forms a nucleic acid. So that's how a nucleic acid is formed. Now this nucleic acid could either be a DNA or an RNA. Now if it is an RNA, it is usually is formed by ribose, okay. So in case if it is a DNA, DNA is formed by 2-deoxyribose. So let me show you that. So if it is DNA, it is formed by 2-deoxyribose. Okay guys. Now when it comes to DNA, there are 4 bases which are mainly involved. Observe this carefully, which is adenine, guanine, cytidine and thymine. So AGCT, so that's how you can remember, AGCT are the bases which are involved in DNA usually, okay. But when it comes to RNA, we would have these 4 now. So we will have AGCU, okay. So the only difference here is DNA will have thymine as one of the bases, while RNA will have uracil as one of the bases. So AGCT and AGCU is the way to remember. DNA is AGCT and RNA is AGCU forms. Okay, this has been asked in competitive exams, so please remember. DNA will have these 4 bases: AGCT, RNA will have AGCU. These are the 4 bases which you can have. Okay, so in case of DNA, if you observe, right, what we have learned that the linkage between sugar and base formed is like an acetate form. Observe carefully here now, this carbon, oxygen and there is no OH group here, right? If it was OH, it would have been hemiacetal, but instead of OH, it is got connected with this nitrogen. So check out the bond carefully. So this carbon connected with nitrogen, oxygen and some R group. So it is making some kind of an acetal form, right? So both in DNA and RNA, we will find this linkage as an acetal form, okay? So which one of the oxygen, instead of one of the oxygen atom, now it is connected with nitrogen atom, but the form is same as that of acetal, okay? Now check the next thing. If OH is absent or that's 2 prime or that is 2 dash, then we call it as DNA, correct? This is the DNA part, okay, because it is deoxyribose. If OH is present, then we call it as RNA, correct? So that is ribose only, right? So you can generally see that this is the base. Now base, when it is connected, remember when you have a linkage here with the base, we used to call it as glycosidic linkage, and it is connected from beta position. I mean, OH was above the plane, above vertically pointing up, so that is beta, and it is giving a glycosidic linkage. So I will call it as beta glycosidic linkage with base. And if this polymerization continues, see that third 3 prime or 3 dash and 5 prime or 5 dash, if this combination keeps happening from 3 to 5, 3 to 5 and it continues, you will get a polymerized chain, and that is what is called as DNA. I suppose you understood that, everyone. So this phospho group starts at 3 bar and ends at 5 bar, and if it keeps continuing, right, an alternative structure of diether which keeps continuing at 3 cross and 5 cross, right, that would give us you know, polynucleic acid, which is DNA, okay. Now DNA is also known as a double helical structure because DNA does not be found as a single strand, it is always found as a double strand. See like this, if you can see, it is a double strand, which means this is one strand and this is the other strand, and both of these strands are connected by each other through hydrogen bondings, correct? Now on the strands, if you check out, there is a phosphate and sugar which is present on these rails, and these are called rugs as in the logs which are normally found between the rails, correct? So which means there have two formations here, correct? So two helix are bound by hydrogen bondings, okay, and we will have phosphates as well as uh, sugars as the rails and bases as the rugs, correct? So you can see that sugar and phosphates appear like rails and bases appears like rungs, correct? Now complementary bases means in DNA, bases do not randomly pair up with each other, but they have fixed pairs, and these pairs will only form double strands. Understand that carefully. What does that mean? See, one base will complement with the other base. For example, when it comes to adenine, it will only complement with guanine, okay, right? Sorry, when it comes to adenine, it will only complement with thymine, while guanine will only complement with cytosine. If you recall, just check it out, adenine and guanine were purine derivatives and thymine and cytosine were pyrimidine derivatives. So purine and pyrimidine derivative is what you will see as a complementary basis. That too, 80GC, got it? So that's how you can check it out. I usually remember this as 80 as in at, GC as in general championships, okay. So at general championships, at as in adenine, thymine, and GC as in guanine combined with cytosine. Okay guys, I hope you understood that. So that is how the DNA is normally connected here, okay. So remember that thymine is always going to be paired up with adenine and vice versa. This is the thymine form, okay, with sugar on this end and phosphate would be connected to that sugar. Next, if you see adenine, sugar on this end and phosphate would be connected somewhere here. Now you see the connection, how the hydrogen bondings they would have, see the hydrogen bondings. So that's how they form the double strand. I hope you understood that. So see, T always complements with A. Similarly, let me show you the other complementation, which is cytosine and guanine. Cytosine, see this side here we have two nitrogens with two hydrogens, and guanine here we have oxygen and hydrogen. So these two will have hydrogen bondings, and here also O and H will have hydrogen bond. And here the connection is between carbon, the bonding is between cytosine and guanine. So they would be complementing each other. I hope that is clear. Okay. So when it comes to hydrolysis of nucleic acid, if you hydrolyze the nucleic acid, then we will get the sugar back, base back and phosphoric acid back. So all will be separated out. Okay. Now when it comes to DNA, we will have two kinds, primary and secondary. Now when it comes to primary, it is just the base sequence, which means the rails we were talking about. Okay, phosphate and sugar combined is giving us a base sequence. Secondary means it is a double helix. So we have seen the structure earlier, it had a double helix structure, right? And when it comes to RNA, RNA will have three kinds here, primary, secondary and tertiary. Okay. The secondary would have alpha helix going through right-handed helix, beta helix making a left-handed helix. Not in the syllabus, all you need to know is just RNA is categorized into primary, secondary and tertiary. That's all it is. See, generally tertiary, if you check out, it will have single strand that may be rewind over itself. Okay. And RNA has three parts, students. One is messenger, ribosomal and transfer RNA. It is represented as mRNA, okay, tRNA, got it, and rRNA. So these are the three RNA kinds which you will see. Basically different functions it has. mRNA has a different function, rRNA has a different function and tRNA has different function. I hope you understood this. Okay, so that's it from nucleic acid, students.